what we are going to do today can be seen on this slide that I'm sharing now with you. And what we have is we'll do a quick warm-up exercise to just get MATLAB up and running and get into this mood of using code to express our thoughts. And we look at the main example of this session, which is the Petri formalism to set up mechanisms to simulate biological happenings, in particular reactions. And as you will see, these can be both molecular reactions, but also reactions between organisms, and therefore population models fall also under this category. And once we have looked at the formal part of that Petri net approach, we'll then end the session with an epidemic model to see how that then works as an application. Maybe before I go to this, I want to alert you that the code for today's session is on the Moodle page for the seismic course. And if you know the online tutorials button at the top of the landing page, if you just scroll down to where it says vectors and matrices, like what I have here now, then vectors and matrices, which is now at the top, the very first link under this heading of session number 4 is linked to the MATLAB code. So whatever I do here will be in a very similar way also written there in case you want to read it up later, or if you are just can't be bothered to copy it from the screen while the video runs. Right. And once we have this, let's go and bring up MATLAB. I think I want to stop sharing my video now, so you have more space on the screen to watch what I'm doing here. Let's see, let's start a new script as usual. Can also save that script under some name. Test. This doesn't really matter what it is. So what we'll do first, we'll get set up a matrix. We'll look at two things. One thing is we have seen before in the network, in the context of biological network matrices, that we can use random number generators to create matrices. So we'll do that again. And the first thing I will show you how to control the random numbers that are generated, which is a very simple trick, but nevertheless useful. And later we'll look at how there are two different ways of addressing the entries in a matrix. So if maybe you remember, we'll have some convention in programming that we don't like so much to use integer numbers or parameter numbers within the code. Would like to define them beforehand. So if I want to have a matrix that is 5 by 5, I can define the matrix size or mat size as with this number, and then I define my matrix. And I use one thing that we have seen before, namely a function called randi. And in this case, I'm creating random numbers, say between -2 and +2. And the reason is I'm thinking of stoichiometry matrices, and stoichiometry matrices typically have numbers that are +1, -1, +2, -2, but not much other than that. And the rest will be zeros. So we have those matrix elements set up, and then we can say, and we want that to be the size that I have defined before, namely mat size. So it will be a 5 by 5 matrix. If I adjust my command window and I enter this code. So you can see there's a matrix that has those entries. However, it's chosen randomly. So if I now keep executing the code every time, maybe I make a little click to it. Can you hear that? So every time I press enter, and the components in that matrix will be different. So the question is there a way of creating matrices that have a specified set of random entries, so randomly created, but every time the same numbers, such that they are reproducible? For example, if you want to use that in a report or in a paper, you would like to share code that others can reproduce to the exact same structure that you used here. So yes, there's a trick, and the way to do it in MATLAB is to set a seed, a so-called seed. Here is just an integer number, and I set it to 1, and then use this seed to prime or initialize the random number generated. So we call the random number generated the function RNG, but we use it with seed. So that's a seeded random number generator. And once we have done that, we define, so we just put it in, assign it to a variable which is called S, namely this sRNG. And once we have set this, we can then make a new cell. Then when we want to call our random number generator, we can do what we did before. This is every time different numbers, but now I change that and I add, before the call of the random number generator, I add RNG of S. So S defined with that specific number, in this case 1, you can use any integer. And in this case, unrecognized number S. So I forgot to initialize it. So now it's initialized. And once I use it, I have the random number seeded. And when I now keep executing the code over and over again, it will give me the exact same random numbers. So it's just a sort of a trick, but to remind us that random number generators are, of course, algorithms in the background that run according to some rule. And if you understand the rule, you can also use that rule, like in this case, to produce the exact same output that you want. And another quick thing to look at is we have seen that now that we have a matrix, it's called my matrix. So use that code here. So that will give me that matrix here. We know also how to access elements of that matrix. Or I guess you know. So if we use the round parentheses and we specify, for example, an element in the second row, and we want the first column of that matrix. So second row, first column would be number 1. So if we execute this code, we will see the answer in the command window, which is 1, right? And that means this is a convention to call that matrix by specifying row followed by specifying column. There's another way to access the matrix, which we can also do, and that is the same my matrix, but just use a single number. And when we do that, in this case, what you see is actually the exact same element. And so I just wanted to highlight this. There's also a way to access every matrix element with a single number, except that single number, the largest of which is 25. And this is a different number, it's now 2. And the reason we have got 25 is that we have now accessed this in the convention column by column counting from 1 to the last element. So the first element, number 1, is here. Second element, number 2, is here. Third element, number 3 is here. Then we have 5 elements. Element number 6 will be second column, first row, and so on all the way down to element number 25, which is my matrix 5, 5. This is important to know, and that outcome can be differing depending on how you call an element in a matrix, and it's confusing if you're not aware of these two ways of accessing features. Right, I think that's sufficient for an introduction. Let's go close this file, clear everything. Clear will just clear the workspace, and CLC will clear what is written in the command window. Okay, so let's go back to the main topic of this session, which is the Petri net. So if you have looked at it, you know all those things are somewhere. Just copy it from the materials. What you see in the left-hand side, on the right-hand side, on the left-hand side is a picture of the very Carl Adam Petri, who introduced this kind of formalism. And the way it was introduced was to get a very convenient way of setting up matrices that allow the setting up and simulation of models of biochemical reactions, be it molecular or population models. And so I don't explain this. Of course, this is in the materials. But what we are going to do is we're going to look at one example of how to work with this and just go through the components that are important for each step in this formalism. So this example is not in the materials. It's just an example reaction. I took some reaction, which is PO reaction. That's short for peroxidase oxidase reaction. It's catalyzed by an enzyme called peroxidase. And it consists in taking hydrogen peroxide, here somewhere on the left-hand side, in an acidic medium in the presence of NADH, so the reduced form NADH. It will convert it to water, to molecules of water, and it will give us the oxidized form NAD plus of that co-substrate. And it's called the PO reaction because in addition to this one here, which is the peroxidase reaction, there's also an oxidase reaction, which is very much related to this, but which actually takes molecular oxygen. So the function of this is probably to get rid of the hydrogen peroxide, to detoxify the cell interior if there's too much reactive oxygen species accumulated. So now if we have this set up from this, putting the reaction in place in this way, we can immediately within the Petri net formalism specify that what we have here is a single reaction. So one instruction of an action that takes place in that model that we want to set up. And it involves just counting 1, 2, 3, 4, 5 different species. And then if you remember the convention that leads to the creation of the reaction matrix, that translates into a reaction matrix with 1 row and 5 columns. So if we have this done here, now that's all that we need, we can now proceed and try to set up this thing in our MATLAB environment. So I go back, I create a new script, and I copy just what I had here. I copy that into MATLAB, and of course I need to make that a comment so it's not mistakenly interpreted as code. So this is what we have so far. So how can we go? We can create our species vector, and species vector, you can put it, if you just want to have the names, So just conveniently, so to have a reminder of the species that are there and also the order in which they are set up, then we would have to put them within curly brackets and quotes for each of the species that we have. So we call the first one H plus, so these are just names that I give to them. H2O2, and the third was, what was that? The reduced form NADH. And then we have, of course, water. What do we call that? H2O, and we have NAD plus. Let's call it like this. So we can execute the code, and you can see it creates a cell array, 1 row, 5 species. So how do we get to the goal of our modeling? What is the goal of our modeling? It's actually to get the stoichiometry matrix. We want to run the stoichiometry matrix. And the way to set up the stoichiometry matrix, if you have looked in how it's done in the materials, we're setting up an input matrix and an output matrix, and then we subtract the output minus the input, and we get the reaction matrix inverted and get the stoichiometry matrix. So that's just in a nutshell. Let's just do that step by step. We'll later have a quicker version. No, and that's not a species matrix. This is just an array. So we use square brackets. So what is the input? If we only need one reaction. So what is the input to the reaction? We can see from the top here, we need one molecule, if you like, of hydrogen ion, one of hydrogen peroxide, and one of NADH. But for the reaction to take place, we don't need any of the products. They will just be produced, but as an input, they get indicated by 0 because we don't need them for the reaction to take place. So that's our input vector. Similarly, we can define the output vector, and we have that in square brackets again. So we have none of the reactants as an output, but we have product 1 and product 2, and I think that's all. So if we execute the code, we can see immediately below that's how it's done. So put semicolon here, so we don't need to see that again, but we now can proceed to create our reaction matrix. And conveniently, that is output minus input. So if we execute the code, we immediately see this is our reaction matrix here. And from the reaction matrix, we can create a stoichiometry matrix S, short for stoichiometry, by taking that reaction matrix and putting the apostrophe next to it, which will just invert it and which will transform the row vector into a column vector. So of course that same thing will apply to matrices. It's just a very simple thing in this case. So now that we have our stoichiometry matrix defined, we can go and do something which is a bit more complex. And what we want to have a look at the entities called invariants. So there's a P invariant and a T invariant. Let's have a look at the P invariant. And we have to remember for this, and I typically have a look at some cheat sheet or something to remember what that actually means, to not confuse the two with each other. So the so-called P invariant, for some reason that is outside of biology, indicates the so-called conserved moieties. Within biochemistry, we call that conserved moieties. For example, couples like NADH, NAD+, or ATP, ADP, and so on. So molecules that are being recycled in a circular fashion and that therefore don't change the number that is the sum of the two together. And so a conserved moiety is a combination of molecules that, if you sort of add them or subtract them or whatever you want to do together, it's a combination of those molecules that will remain unchanged under the action of that model. So even if those reactions run and they take some substrates and they produce some products, nevertheless, the conserved moieties under this model is a number that will remain unchanged. So it's a useful thing to have, something that seems to be very simple now, but might be very difficult once you have a big model. So the way to do it is you call the function null for the so-called null space of a matrix, and you apply that to any of either the reaction or the stoichiometry matrix. So if you want to have the P invariant, you need to apply that to the reaction matrix. So you get the null space of the reaction matrix, and you say you want to see the real parts only of them. And then the output is 4 columns. So means there are 4 conservations, 4 ways to combine molecules whose number under the action of this reaction will not change. And so one of the things is the sum of the first and the second, or the difference actually, because one is negative. So the way to read it is to go, which I can't highlight here, is to go column-wise. So we have 4 conserved moieties, and the way to read that, the first column is to read, you take the number of the molecules of the first species negatively, and then you add to it the number of the molecules of the second species. So if we have our cell array up here, so that hydrogen ion and hydrogen peroxide, if you take negatively the number of hydrogen ions and add to that the number of molecules of hydrogen peroxide, that number over the course of the reaction will not change. And of course you can guess why that is, because both are consumed. So if one is negative and one is positive, whatever their initial numbers are, that sum will not change. And the same goes for the others. So just to remind us, that would have been the P invariance. So let's look in a similar way, commented at the T variance. So we get this from what? From the null space applied to the stoichiometry matrix, the same, only real parts. Take the first one away. And what we can see here is that the answer to this question now, the null space of the stoichiometry matrix in this special case with only one reaction, is empty. Now, what was it? T invariant is a combination of reactions. Now, it's not about species, but about reactions, a combination of reactions that doesn't alter particle numbers. So if we get an empty row vector here, means there are no T invariants. There isn't any combination of reactions that will not lead to changes in particle numbers, which is a bit trivial for a single reaction that is directed, namely goes only in the forward direction. Of course, each of the numbers of species will have to change. The substrates are being degraded or decreased, and products are being increased, product numbers are being increased. So of course there isn't a way to preserve numbers because we have only one reaction. But in the model, in the examples that are in the materials, you'll see examples how that leads to combination of reactions that leave the particle numbers unchanged. And these are in a sense important because, of course, not changing particle numbers means there's a change over time, so something that varies. But if there are numbers that are unchanged, something that is stable, that is sort of a prediction of a sort of maybe you remember an equilibrium state where things are unchanged over time. And this will then apply also to reactions that are taking place far from equilibrium, so non-equilibrium, which is the generic case in biological systems. So even though there's a lot of reactions going on, it's still possible that some of the particle numbers don't change at all according to the model, or in reality then change only very slowly, which is the typical case in experiments. Right, so that was this difficult concept of invariance. And if I just make a little bit of space here, finally, we want to look at sort of a simulation of that model. So what do you need to simulate such a model? First of all, you need to define a rate constant. Right? A rate constant means at which speed does the reaction that you have occur. And if you have multiple reactions, you would have to define that for each reaction such that you have relative speeds, which reaction is faster, which is slower. So if I just follow the convention in the materials, put that into a vector, in this case we have only one. I take an arbitrary example now. We just assume that this reaction that we have, the PO reaction, takes place at two reactions happening per time unit. So this is one way of setting up. You have that vector which contains the numbers of events that happen per time unit. And of course, that would contain more elements if we had more reactions. In this case, a single reaction, so it happens only once. And then the change that actually happens is, of course, we know the change that happens depends on the stoichiometry matrix. And that's the stoichiometry, that's how much of each you get, how much of each you take or produce in every run of the whole model. And you can get then the correct changes according to the time constants if you multiply that with that value u. So you can see in this case it has just doubled our number because it means each reaction as generated by that stoichiometry matrix S will take away one molecule according to the way we set it up of each of the substrates and create one molecule of each of the, each of the products. Is that actually true, or am I doing? Aha, and now I can see that I have done something wrong previously, so I need to change that immediately. What's wrong with this reaction here? Can you see that I've actually missed out a factor? As it says that actually from hydrogen peroxide and hydrogen ions, we get two, not one molecule of water. So if we wanted to correct that, and now we have seen it, that is the species number 4. Then it's not one of those, it's actually two of those. And if I run that code again and look at our stoichiometry matrix, has now slightly changed, and it now involves the fact that we get two molecules of water in each of the reactions. So the output of the null space has not changed. It's still an irreversible reaction going only one direction. So there still isn't any T, and it's called variant. It's called T invariant. There's still no T invariant, of course. And if we now go, we get now the corrected particle numbers, which is -2 for each of the substrates per time unit, and 4 molecules of water, but 2 of the NAD plus the oxidized version of that co-substrate. So, but our original goal was not so much to see that this is going to happen, but to actually check, but to actually set up some mechanism that allows us to simulate the model. So what do we need to simulate the model? We need to define the initial particle numbers. We need to set up a model with specified numbers of how many particles of each of those species that we want to simulate do we have present at a given time point. So one thing to do that is to, sort of not plus, but to define the initial particle number as an array. And because we want to apply that to the stoichiometry matrix, this array needs to be put up as a column vector. So let's assume 10 particles of each of the substrates, just arbitrarily. It's actually any number will do, but so we have 10 hydrogen ions, 10 hydrogen peroxide, and 10 of NADH. And then initially no water and initially no NAD plus will just be a 0, 0 vector. So we have the initial particle number, and you can see it's nicely set up as a column vector. And then in order to proceed with these initial particles and make a simulation, how do we do a simulation? We take that initial particle number and we say, let's assume that 1 unit of time passes. And then we know per unit of time we have a change of the particle numbers that follows S times U, as we have just checked previously. And so we need to use that product S times U to update the particle number. So if we do that once, you can see the result in the command window. Then we are left with 8 of the substrate molecules, each of them 8 because 2 reactions per time unit have occurred. And so that brought us down from 10 to 9 to 8. And we have 2 of the NAD plus, and because water is created at 2 molecules per reaction, then that will bring 4 water molecules. And of course, if you now say, I want to make a simulation for a certain amount of time, then a certain amount of time you have to define that. Say time or duration or whatever that is. You say maybe 5 units of time. You want to have that reaction run over 5 units of time. Then you need to do as many changes as you have units of time. So multiply that, what you have, that instruction to change per unit of time multiplied with the amount of time units that you want to simulate. So in this case, if we want to simulate 5 units of time and execute the code, we can see in the command window the result. Because 2 molecules of each of the substrates are being consumed. And we started with 10 of each of them, and after 5 time units, molecules are consumed. So we end up with 0 substrates in each of the species. And of the products, we have 10 of the oxidized form NAD+, and we have 20 molecules of water in this specific example. So it's now a very simple thing, but what you can see that this is a very explicit mechanism that tells you once you have set up your stoichiometry matrix and defined your reaction rate constants and the amount of time you want to simulate, you can get the result right away. Right. So that was what I wanted to do as an example. So we've now done the full cycle. We've sort of set up our reaction, created the species vector, then defined the actions and defined the species that are involved in this, looked at some features of the resulting stoichiometry matrix and reaction matrix by means of these invariants, and then set it up with rate constant such that we can actually simulate what's going to happen. And this is sort of the heart, the essence of what is happening when you do computational simulations of models. Right, For the last part of this morning, let's look at a specific model. Let's look at the epidemics model. This is a model that we have, actually is one of our final assignments. So epidemics model means something that tries to simulate the spread of an infectious disease, and it looks at changes, not so much at molecules, but it looks at changes of, so if you like, phenotypic changes in populations. And that can be, of course, bacteria or mice in the experimental condition, or it can be humans in the case you want to have a look at infectious diseases. So the epidemics model that we have here, that is the basis of one of the final projects of this course. I'll show you quickly how it is set up. So I explain the assumptions that go into the setting up of the model. Then we'll look at how to create stoichiometry matrix, just as we did now. We can have a quick look at TNP invariance. And I'll show you an example of a simulation of such a model where you can see, I think, something that is not really done very explicitly in the material for this course, but that this directly translates into making predictions from the assumptions, which is the core of computational modeling. Right, so we need to go back to MATLAB. Let's just close this. Don't need to change. Clear. CLC, get rid of everything. So that was done to make our workspace empty. So none of the previous variables is there anymore. That's particularly important because if we define our stoichiometry matrix using like the capital letter S, then we avoid to have any confusion with previously defined S's. So what we want to do is to start a new script, and we can save that at, it's called the SIR model. And we'll just call it the SIR model something, just give it a name. And we'll try to implement it very similar by defining, in this case, species or components involved in that model, and also the actions taking place. So let's first define the actions, and then the species that we have. Species, let's misspell species. So define those two things, and for the ability to memorize it, let's do that similarly to how we did it before in a nice array that will help us to check each time in which order we have defined them. So the actions that we have, according to the model that I just showed before, but I'll do it verbally now, and that we have a couple of different species that interact with each other in the following way. So we have a susceptible population. This is all the individuals in a population that have not yet had a certain disease and that are susceptible to infection. And then we assume that we have at least some infected people that exist in that population. So somebody has got some infection, not mentioning any names here, and that there's an interaction that if a susceptible person meets an infected person, then what happens according to this model is that they form, so make a symbolic arrow here, that they form a pair, a contact pair, so to speak. So they get in close contact, not saying for long or for short, just they form a contact pair, means they allow for the infection to pass, say for a viral or bacterial infection to pass from one to the other. So the formation of the contact pair is our first action in that model. And then what can happen to this contact pair? So this contact pair then, Si is the contact pair, and this contact pair can then decompose, so they go apart again. And what happens after they go apart? They will produce effectively two infected individuals, namely the first one was already infected, remains infected, but the second one that was susceptible has received an infectious pathogen and is now also infected. So we get two infected once the pair dissociates. And our third action that we have, everybody who is infected hopefully has a good course of the disease, but goes through the disease and then recovers after some time. So we specify that by assuming that an infected individual converts into a recovered individual R. And that's all the assumptions that we have for that simple model here. All right, so a susceptible person can get infected when they meet somebody who is infected. The contact pair dissociates and leaves everybody involved infected. And at some point after you are infected, you will just turn into a recovered person again, means you will just go back to healthy, go back to normal. So that's a benevolent disease model. And then we can set up our curly brackets, we can set up our species vector. So we have a species S, which are our susceptible individuals. We have the infected individuals. And of course, we have the contact pair Si, which is now a different entity, so to speak. And we have the recovered. And if you like, according to what we did previously, would be actually nicer to set them up as a column vector to be consistent with previously. So we have three actions or three reactions, if you like, and we have four species that join that model. So if we execute it, so we have two cell arrays that contain these pieces of information. We just suppress any further output by putting a semicolon there. And we can now go to define our stoichiometry matrix directly. So we don't do input and output and subtract them and invert the reaction matrix. We just set up directly our stoichiometry matrix for this simple model. So we set it up initially with 0, and then we populate it with the assumptions that we have made. So let's have a look. What do we have? We have 4 species, so that's 4 rows for the stoichiometry matrix, and we have 3 actions or reactions, so it's a 4x3 matrix, and let's populate it. Let's see what happens to all the species in the first action or reaction. So it means we now have consequences of the first, so the column here is the first reaction, and now we need to say what happens to the first species. It's called S, so that will disappear when they make a contact pair. S plus I goes to Si, so it will change by -1. And then very similarly, 2 in the first reaction will be the same. That's the I variable, the infected will also be taken away according to this model. We'll come to that setup later. So how many do we take? I take away 1, right, in that first action. So next thing is the third species, which is the Si contact pair. In the course of that first reaction will be +1. So this is all that we have from that first reaction. Now let's do the part that has to do with the second reaction. So the contact pair Si goes to 2I. So, and all of this is in the second reaction. So we can define, so we have two things happening in the second reaction. The first is what happens to Si. So Si is our third species, right? So we are here in this second reaction. So Si is our third species. So the third species will be reduced by -1 because one contact pair will be taken away. And then we'll get from this 2i, so I is our second species, but we'll get 2 of them. So we'll get 2 out of those. And that brings us to the very last reaction, which is fairly simple. And I just complete that. So in the last reaction, an infected goes to be recovered. So the infected was 2. In the third reaction will be -1. So will be 1 goes away or 1 disappears. And that will give us a recovered, that is the fourth species. In the course of that third reaction will be, of course, +1 then, because for each that disappears, 1 recovered will appear. And if we have done everything correctly, we end up with our stoichiometry matrix. You can see it below here in the command window. So we have our stoichiometry matrix now set up. And you can have a look at this. As you saw, I immediately abbreviated. Once you are a little bit fluent in this formalism, you don't go through the input and output necessarily unless you want to make very sure, or you need the reaction matrix for some other purpose. If it's just for the setting up of the model, that is the quicker way to do it. And as you can see, if you have your definitions according to this convention that we have set up, this can also be done automatically. I'm trying to open that code. So I have it now. Just copy and paste, and let's do that here. Let's have a look at how this code looks, and then we execute it. And so now I have used the convention that we have done before, namely defining the species vector, which is here, where we have defined that as results. And I say I start with a population of 20,000 susceptible, 11 infected. So some infected are in that population. And no contact pairs, no recovered person initially. And then once we have that, we have set up the relative rates of those reactions. So I'm just putting in some arbitrary numbers. So remember, we have 3 actions that are taking place. So we put 3 numbers, which are the 3 rate constants or the relative speeds of those reactions. And then I have successively for each of the steps created a result vector for the amount of, in this case, 20,000 steps or 20,000 time units, if you like, is then I'm going to proceed to simulate over a long time. And what I have done here, very much in the spirit that you learned in the very first session of this course, made a for loop to collect all those results and then plot all those results. And if we've done everything right, that should give a nice output. So I execute the code, and what we get is a simulation of that very simple or simplistic SIR model of infectious diseases, where we can see the outbreak of a pandemic, so to speak, or the outbreak of an epidemic, maybe less dramatically, where the initial population of susceptible people indicated by the blue curve goes down. Means why is that? Of course, because more and more people get infected. So the red curve shows us how the number of infected people goes up. And then the number of contact pairs stays close or near to 0 for all the time, because that's something they produce, but they also split. And such there's no, at any time point, there isn't a large number of contact pairs, which is, of course, a fairly good thing. And then once you have the recovery process set in, so people get healthy again, that magenta curve at the bottom, then is the accumulation of recovered people, those that have had the disease and are now forming a new population, so to speak, those with antibodies that are no longer susceptible to it anymore. Right, that's it. That's what I wanted to show you. So this whole thing can be set up in using the Petri net formalism. You need to be able to handle vectors and matrices in a certain way. And then hopefully, if everything is right, you get possibility to make simulations such as those that I've done here. Good success with your assignments. We have set up online tutors that will look at the assignments as usual. So if you have questions, please put them into the forum posts for this session, and we'll try to get back to you and give you feedback on what you have done by request. Our next session will be in 2 weeks' time on Functions in Calculus 1, where we'll go a little bit into the details of the using a mathematical way of expressing different functions and how to implement them to make the model more realistic. There's one thing that I have sort of avoided in the picture that is shown here on the screen is that if you keep simulating that for a longer time, that blue curve actually here crosses zero and goes to negative species. So there would be negative numbers of susceptible people, which is of course nonsense. But in order to amend that, we'll need to have proper mathematical functions, not that primitive as we have it here, but it's a little bit more sophisticated. Right, so that's it for today. Thank you very much for watching. If you like, see you in 2 weeks, and otherwise enjoy working with the course and coding in MATLAB.